I'm Zane Grabau. I'm a nematologist at the University of Florida. I'm based in Gainesville, but I cover row crops and horticulture crops throughout North Florida. I'm here at the NFREC in Marianna today, giving an update on nematode management in row crops. So I'm going to give a short demonstration of soil sampling for nematodes. This is one of the important steps in nematode management. In order to select a proper nematode management option, it's important to know if you have nematodes in the field and what nematode you have, because your management choices are going to vary based on what nematode is present in the field. Some options as far as rotation crops or resistant cultivars work against particular nematodes and not other nematodes, so it's important to know what nematode you have in the field. So what we always want to do when we're sampling for nematodes is to always collect a soil sample. You can also collect root samples or send pictures of that as well, but that's kind of a bonus. The reason we want to collect soil samples is because some nematodes are not found in the roots, they're only found in the soil. So to demonstrate taking soil sample, you have here a soil probe. Any kind of tool that's going to get you down to about 8 inches or so in the soil is going to work fine. Probably most people are relatively familiar with taking soil samples for nutrients and other things. Kind of the two key things as far as sampling for nematodes are that we want to make sure that we're getting close to the plant roots, because that's where the nematodes are most concentrated, around the plants. The other thing is handling the samples. Unlike soil samples for nutrients, we don't want to let these samples dry out. So it's important after you collect them to store them in a relatively cool place out of the sun, and when you get back to your lab or wherever you're storing them at, to put them in the refrigerator. So collecting the soil sample. Again, with the probe close to the plant roots, trying to get down about the whole length of the probe. And we're going to take about 12 cores or so throughout the infected area. So you can see here, we've picked out an area of cotton that looks like it could potentially have some nematodes in this area. You see it kind of in the background, we have green, relatively tall cotton, and this patch that I'm standing in right here has matured a little bit earlier. It's quite short. This would be an example of one of the symptoms you're looking for as far as where you would target to sample for nematodes. This is a great time of year to be sampling for nematodes because you still have the plants in the ground where the nematodes populations are going to increase on those plants. So populations are likely to be high at this time. So this is a good time to be sampling in order to guide your management for the next year. It's too late in this year to do anything as far as management, but this is a good time as far as looking for future years. So again, we want about 12 cores or so. Sampling particularly in the areas that you think may potentially have some nematode problems. And the bigger the area that you're seeing problems, the larger area that you want to cover in your sample, up to about 5 acres for a sample. But if it's a smaller patch, you may sample a smaller area. It can also be useful to collect a separate sample from a healthy area so that you have comparison of what the nematode counts are in a healthy area versus a diseased area. Okay, so I've collected my samples now. I'm going to mix up the soil sample. And Always important to label your bag with the location of the sample. And after we've mixed up the sample, we can put it in the bag. That's about the volume that we want. You can get by with maybe a little bit less than that. I'm going to seal this up, and then it would go in a cooler in my truck until it gets back to a refrigerator. So one thing I'll also mention, University of Florida has a nematode assay lab that runs these samples. If you are looking to diagnose a problem in a commercial field and you work with an extension agent in row crops, the Nematode Assay Lab does run those samples for free as long as it's a limited number of samples, looking at diagnosis again in a commercial field. So other than soil samples, the other thing we want to look for as well when trying to diagnose nematode problems is to take a look at the roots. So I'm going to grab my shovel here. So dig up roots and take a look and see if there's any potential symptoms of nematode infection on the roots. In this particular area, I haven't seen any symptoms, but some of the very common symptoms would be galling, which are abnormal knots or growths on the roots is a diagnostic symptom for root knot nematode. General stunting of the roots is a common symptom, pruning of lateral roots. So taking a look at these roots and using EDIS guides to, if you're not sure what symptoms you should be looking for in a particular crop, can be useful. So like I did with the soil samples, I want to take a look at a few different roots. So again, these roots to me, I don't see any obvious symptoms of nematodes on them. No galling, no obvious stunting that would be typical of nematodes. But if you do see symptoms, it's important to take note of those when you send in your samples. If you're just sending a soil sample, you can also send in a sample of the roots as well when you send in a sample to the Nematode Assay Lab. Another thing that's very useful is taking pictures both of the roots and also of the symptoms in the field. They can give us an idea when a specialist such as myself is trying to diagnose the problem, what the symptoms you're actually seeing in the field are and if they're consistent with a nematode infection. So today I'm going to give an update on nematode management in row crops. The three crops I'm going to be talking about today are corn, cotton, and peanut. So just a kind of a reminder of general nematode management. We rely on three different management practices, primarily resistant cultivars, crop rotation, and nematicide application. So these will come up again throughout the talk. So first, in corn, I want to talk about a couple of trials that we have conducted over the past couple of years. The first one focuses on nematicides. In corn, the two nematodes that we deal with most, that are most problematic, are sting nematode and stubby root nematode. And these were the two nematodes that were most prevalent in the nematicide trial. This trial was conducted at the Hastings Research Center. We did it in 2019 and 2020. We were looking at both some older nematicides in corn. The primary option that's typically used is Counter, which is a granular nematicide that's applied in furrow. We also looked at a couple of different rates of telone applied in the row at 3 gallons and 6 gallons per acre. And then we also looked at a relatively new product that's been around for a couple years in cotton and peanut, but just got labeled this year in corn. Currently what's labeled in corn is called Propulse. The active ingredient in that is fluopyram, which is the same active ingredient in Vellum Total, which is the product labeled for cotton and peanut. So we compared those different nematicides in this trial. And the Propulse, like the counter, is applied as an in-furrow product, except that the Propulse is a liquid rather than a granular. So big picture, what we saw in this trial was that counter in general performed the best among the nematicides. It was the most consistent product against both sting and stubby root nematode. Talon was good against stubby root nematode in 2020, but in 2019 it was relatively poor as far as managing populations of that nematode, which is not incredibly surprising. Based on research in other crops at Hastings, we know that fumigant nematicides, for whatever reason, tend to not control stubby root nematodes incredibly well, even though in general, Talon and other fumigant nematicides tend to work very well against most nematodes. Vellum, or Propulse, I should say, was generally intermediate against both sting and stubby root nematodes. As far as the yield, in 2020, counter performed the best out of any of the products. We had a 60 bushel increase over untreated control. Counter was 35 bushels per acre better than our Talon or our Propulse treatments, and the Talon and the Propulse treatments had a 25 bushel per acre increase over untreated control. In 2019, we had a mechanical issue with our planter for some of our treatments, so we only had yield data for our counter and Propulse, but in that year, counter outperformed Propulse by 25 bushels per acre. So that's an overview of that nematicide trial in corn. Second trial I want to talk about briefly is a cover crop trial. So this trial was focused more on silage corn rather than grain corn, done in collaboration with Marcelo Walu and Jose Dubois. This trial was conducted at Citra, and we're just now finishing the first cycle of that trial. What we did is we planted late summer or fall cover crops after a planting of spring corn. So the crops that we looked at were sun hemp, pearl millet, both a fertilized and an unfertilized option as a check. And then we also looked at sorghum Sudan grass, and then corn as kind of an option where you're looking at corn on corn on corn. So what we found from that trial is that sun hemp generally worked best for, again, a sting stubby root nematode combination in controlling both of those nematodes. Again, this is just from the first year of data, so we're repeating that trial to get more data on it. We didn't see any statistical differences in our corn yield following those cover crops in the spring, but numerically we had a 40% increase in yield following sun hemp compared to our grasses. So this is typical of what we see in rotating. Typically, again, you're dealing with sting and stubby root nematodes as your most problematic nematodes on corn. And so to break up the problems with those nematodes, what you want to do is get away from grasses. So sun hemp is a legume, and it's a good option as far as getting away from grasses. And your options are going to be a little bit different for cash crops. You're looking more at things like cotton and peanut to break up your rotation if you have a problem with sting and stubby root and you're getting too much corn in your rotation. But again, important to rotate. So moving on to peanut and cotton, I just kind of want to give a brief update on these. Again, reiterate, there's three main options that we're looking at with managing nematodes: resistant cultivars, nematicides, and crop rotation. So in peanut, we really have some very strong resistant cultivars that are available right now. The main one right now is TIF NV, but others also available a little bit older: TIF Guard, Georgia 14N. All of these have a similar parent, and they have very, very strong resistance to root knot nematodes. You plant these varieties in the field that has heavy root knot nematode pressure, they're going to perform about as well as they would in the field that has no root knot nematode pressure. So under very heavy root knot nematode pressure, these are a very good option for preserving yield. One thing they will not do is if you have other nematode problems, sting nematodes, some sting nematodes can affect peanut. Lesion nematode can be a somewhat more minor problem on peanut, but it can cause problems. These varieties are resistant only to root knot nematodes, so they're not going to work against those other nematodes that you may have in the field. So the other option again is nematicide application. So some data we've seen over the past couple of years suggests in a heavy root knot nematode situation that you're probably better off going with a resistant cultivar than a nematicide with a susceptible cultivar. Our options as far as nematicides in peanut really haven't changed much in the last few years, although we are hoping to have some new nematicides labeled in the next few years. Basically, you're looking at Telone as a fumigant. We have Velum Total, which may be having a name change coming up soon. And then Ag Logic are kind of our main options. There may be a few others out there as well, but those are our main options. And again, the last option as far as management in peanut is crop rotation. I'm not going to go through all the options there, but just again, remember, important to rotate your crops. So brief update for cotton. So again, there are resistant varieties available in cotton, and there is some new information on that. One point is that on root knot nematode cultivars, there's a number of them available. What you want to look for in cotton is for cultivars that have double gene resistance as far as root knot nematode. These are going to be much more effective than single gene root knot nematode resistant cultivars. And still, even with a double gene cultivar, you're probably not likely to get quite the same level of resistance as you do in peanut, where it's a near immune, but it's definitely going to give you a bump. The other developing or new management strategy as far as resistant cultivars is that Phytogen is coming out with cultivars that are resistant to reniform nematode. So based on the limited amount of information available so far, these look like they do have some pretty decent efficacy against reniform nematodes. So that's a new tool in the tool belt that should be available coming up in the next years is resistant, reniform nematode resistant cultivars. As far as nematicides, the situation is similar to peanut. We don't really have any new products that have come out in the last couple years, but hopefully in the next couple years we should maybe be seeing some new chemistries that migrate towards cotton, hopefully. The last bit of information that's a little bit different for cotton than it is for peanut is that in cotton we do have some options as far as not just in-furrow or fumigant nematicides, but there are some seed treatments that are available. A number of different seed treatments are available. We have done a number of research studies on a variety of different seed treatments over the years, last few years, and in general, to be honest, we haven't seen a ton of efficacy with the seed treatments. Typically what you're looking for with these is for use in relatively low nematode pressure. Under high nematode pressure, you definitely want to be relying on more of an in-furrow or fumigant nematicide or a resistant cultivar. And then again, just to re-emphasize, our last nematode management option in cotton is crop rotation. So rotate those crops when you can.